Uh, Good morning, everyone. Thanks so much for coming to this talk. And my name is Sijia, and this work is in collaboration with Martín, Neil, and Luisa. So the problem we are considering is that in real life, in real world, there's a lot of problems coming with more than one dimensions. So for example, for traffic, you have a problem of where and when does it happen, and also in epidemic transmissions, there's a problem of when and where and which people are in contact with each other. And then in biology, there's this question of you have genes in the cells, and genes in one cell would interact with each other, and the cells there's a relationships between different cells as well. But however, sometimes with these problems, you only get one set of data. So like for genes and cells, you got one reading for each gene in one cell, and that's it. So our issue would be considering a two-dimensional data. This is in the form of matrix. And we try to infer the structure underlying both different directions in different dimensions. So our assumption is that the structure along the rows and the structure along the columns combined together would be a Cartesian product, and the Cartesian product of the two structures would be the overall structure of the whole data set. So that's an assumption here. And the overall process of this model we're doing is we have some count data, because unfortunately sometimes data don't just come as a continuous data and you just plug them into Gaussian process. But so we have to deal with the count data here, but we still want to use Gaussian distribution, well, because it's mathematically convenient. So what we do here is we do a non-paranormal transformation. Basically, takes the Kendall's tau from the dimension of rows and the Kendall's tau of dimension of columns, and that kind of extracts the information from both directions. And with that information extracted in the form of estimated covariance matrix, here it's called S and T, we then throw it into this objective function, which is a log Gaussian likelihood plus some regularization. And well, the regularization is here because we want to control the sparsity of the problem, so we don't get too complicated a network. And then it goes through the Scalable Bigraphical Lasso algorithm. And voila, there's our networks. Well, I'm afraid there's a bit of math over here, and I'll try to be quick. So the issue here is actually, so Y here is the data set we had, and the assumption is it follows the vectorized version of this data, follows a multivariant normal distribution with a covariance matrix in the form of the inverse of a Cartesian product. And here the matrix psi and the matrix theta represents the inverse covariance matrix on the dimension of rows and on the dimension of columns. And then we got this objective function, log Gaussian likelihood plus some penalties. And then basically get from here, we can get a stationary point focusing on one of the small inverse covariance matrix like psi. And then we consider eigen decomposition. And basically what we did is we rearrange the overall covariance matrix and the overall inverse covariance matrix And so we have an estimation of the overall inverse covariance matrix, and in the form of this, using some of the eigen decomposition, and that will make the calculation a bit easier, because then we got to see to solve each column of the matrix psi. We can just do it by solving this linear equation. And also we reduce the computational complexity and the memory requirements by exploiting the eigen decompositions a bit more. So, well, that's just improves the computational speed and the memory, and reduces the memory requirements. And well, and this is the overall structure of the algorithm. Basically, you do go through each columns of one of the inverse covariance matrix, and then you move to another inverse covariance matrix and update it, and then it just goes through a loop until its own convergence. And here's some results from synthetic data. As you can see, compared with previous methods, our methods are much quicker, where the red line is our method and the blue line is a previous method, while we maintain a good accuracy compared to this other method called Lasso. And we also can get some decent ROC curves and accuracy, TPR and FPR, depends on the penalty parameters we're using. And here's some results from real data. So this is a problem of genes and cells. And on the left-hand side, this is the structure inferred for the cells. And on the right-hand side, this is structure inferred for the genes. But here, we filter the data so that we know there's 3 blocks of different cells, cells in different phases, and the genes are chosen to be more active during mitosis. So it's expected that we found out this third cluster of the cells because all this mitosis was happening in the third phase. And also all the genes are just connected with each other because of their own work together during the mitosis. And so in summary, we exploited the eigenvalue decomposition to present a more efficient version to simultaneously infer the structure in both rows and columns in a data set, and we do it very efficiently. We proposed this using the non-paranormal approach to make it easier to process count data. And well, we have code and data available online, and you are very welcome to read our paper. Thank you so much for your attention.